When I was studying for my HSC, I spent weeks trying to memorize every single step of DNA replication. I thought that's what I needed to do to score a band 6. But when the exam came, I realized I was missing the bigger picture. Now, after years of tutoring students, I've learned the real secret to acing Module 5. Seeing the connections. So once you understand how DNA leads to proteins, how meiosis creates genetic diversity, and how inheritance shapes populations, everything else makes sense. That's what I'm going to show you today. If you want to understand how genetic information is passed from one generation to the next, you need to start with DNA. DNA is the instruction manual for life, storing all of the genetic information that makes you, well, you. So every cell in your body contains the same DNA, yet different cells perform different functions because of how genes are expressed. Understanding DNA structure and replication is foundational because it explains how genetic information is passed down from generation to generation. Let's start with structure. DNA is a double helix, meaning it's made of 2 strands kind of twisted around each other like a spiraled ladder. The rungs of this ladder are made up of nitrogenous bases. Adenine, A, pairs with thymine, T, and cytosine, C, pairs with guanine, G. These base pairing rules are always followed, which is critical for DNA's ability to copy itself accurately. The strands run in opposite directions, something called anti-parallel orientation, which just means one strand is upside down compared to the other. Replication happens before a cell divides, ensuring that each new cell gets an exact copy of DNA. The process starts when helicase, an enzyme, unwinds the double helix and separates the strands. Another enzyme, DNA polymerase, moves along each strand and adds complementary bases to create 2 identical DNA molecules. Because each new DNA molecule keeps one original strand and adds one new strand, this process is called semi-conservative replication. Half old and half new. A smart way to study this is by teaching it to yourself out loud in 3 simple steps. Step 1 is the DNA unwinds. So helicase breaks these hydrogen bonds between bases. Step 2 is that new strands form. So DNA polymerase builds complementary strands. And step 3 is 2 identical molecules are created, each with one original and then one new strand. The reason DNA's ability to replicate is so important is because mistakes, or mutations, can lead to changes in traits, genetic disorders, or even evolution over time. If you understand how DNA stores genetic information, the next step is learning how that information is used to build the proteins that make life possible. DNA on its own doesn't do anything. It's just a set of instructions. The real work happens when those instructions are used to build proteins, which are responsible for almost everything in your body, from muscle movement to immune response. This process happens in 2 major steps: transcription and translation. Step 1, transcription, takes place in the nucleus of the cell. Think of it like copying down a recipe from a cookbook. You don't take the whole book to the kitchen, you just write down the specific set of instructions you need. In the same way, transcription allows the cell to create messenger RNA. We call it mRNA, a copy of a gene which can then leave the nucleus and be used to make a protein. Here's how transcription works. The DNA unwinds, and an enzyme called RNA polymerase binds to a specific gene. RNA polymerase reads one strand of DNA and then builds a complementary strand of mRNA, replacing thymine, which was the T, with uracil, U. The mRNA strand then detaches and then leaves the nucleus, heading to a ribosome where the next step, translation, begins. Now comes translation, which happens at the ribosome, the cell's protein factory. If transcription was like copying down from a recipe, translation is like actually cooking the meal. So the ribosome reads the mRNA in groups of 3 bases called codons, and each of these codons code for a very specific amino acid. Another molecule, transfer RNA, or tRNA, brings the correct amino acids and then links them together to form the polypeptide chain, which then folds into like a functional protein. Okay, so all you need to know is that the changes in this process can lead to genetic disorders, evolution, and even diseases like cancer. If a mutation occurs in DNA, it can alter the mRNA sequence, which can then lead to the wrong amino acid being added to a protein, potentially changing its function. This is where genetics and inheritance starts to come into play, which leads us to the next key topic, how meiosis creates genetic variation. Meiosis is the process that produces gametes, or sperm and egg cells. It's the reason why no 2 people are exactly the same. Unlike mitosis, which creates identical daughter cells, meiosis ensures genetic diversity by having the chromosome number and shuffling genetic material in the process. Meiosis happens in 2 stages, meiosis 1 and meiosis 2. In meiosis 1, homologous chromosomes, so one from each parent, pair up and then exchange the genetic material in a process called crossing over. This is crucial because it introduces new combinations of alleles, mixing up the genetic information so that offspring don't inherit just a direct copy of either parent's genome. These homologous chromosomes separate into different cells, each now containing a unique mix of genetic information. In meiosis 2, sister chromatids of each chromosome are pulled apart, similar to what happens in mitosis. This results in 4 non-identical haploid gametes, each containing half the number of chromosomes as a normal body cell. These 2 key processes that introduce genetic variation during meiosis are crossing over and independent assortment. Crossing over, the first one, happens in prophase 1, when homologous chromosomes swap segments of DNA. This means that even if you inherit the same chromosome from your parents, the version that gets passed on to the next generation is a unique combination of their genes. The second one is independent assortment, which happens in metaphase 1, when chromosomes line up randomly before then being separated. This means that for every homologous pair, there's a 50-50 chance of inheriting either the maternal or paternal chromosome, leading to millions of possible genetic combinations in gametes. The reason meiosis matters so much is because genetic variation is what drives evolution. When populations have more genetic diversity, they're able to adapt to the changes in the environment. If every organism were genetically identical, a single disease could wipe out an entire species. That's why meiosis isn't just important for individuals, it's essential for the survival of entire populations. Next, we'll explore how these genetic variations are inherited and then passed on through generations, which brings us to Mendelian and non-Mendelian inheritance. Mendelian inheritance is based on the work of Gregor Mendel, who discovered the basic principles of heredity by studying pea plants. His experiments revealed that traits are inherited in predictable ways, following the specific patterns based on dominant and recessive alleles. Each organism has 2 copies of every gene, one from each parent. These genes can exist in different forms called alleles. Some alleles are dominant, meaning they will always be expressed if present, while others are recessive, meaning they are only expressed if copies of both genes carry the recessive alleles. For example, if brown eyes, which we'll call capital B, are dominant and blue eyes, which we'll call a lowercase b, are recessive, someone with uppercase B or uppercase B lowercase b will have brown eyes, while only those with lowercase b will have blue eyes. This pattern follows Mendel's law of dominance. Mendel also discovered the other 2 key principles. First of these was the law of segregation. During meiosis, alleles for a trait are separated so that each gamete only gets 1 copy. This ensures genetic variety when fertilization happens. The second one is the law of independent assortment. This basically states that genes for different traits are inherited independently, meaning inheriting one trait, for example, eye color, doesn't influence the inheritance of another, for example, height, as long as the genes are on different chromosomes. However, not all traits follow these simple rules. That's where non-Mendelian inheritance comes in. Codominance is an example of non-Mendelian inheritance where both alleles are expressed equally. For example, this is present in AB blood type, where both the A and the B alleles are dominant. Incomplete dominance is another example of this, which is a blend of both alleles. For example, a red and a white flower can produce pink offspring. Polygenetic inheritance is a third example, where the traits are controlled by multiple genes leading to a wide range of variation. For example, skin color, height, and eye color. And lastly, sex-linked inheritance. Some genes are carried on the X or the Y chromosomes, meaning their inheritance patterns differ based on sex. For example, color blindness and hemophilia, which are more common in males because they only have 1 X chromosome. Understanding when to use Mendelian versus non-Mendelian inheritance is key for HSC exams. Many students lose marks by assuming all inheritance patterns follow simple dominant and recessive rules. Instead, it's important to recognize when traits deviate from Mendelian genetics and applying the correct pattern. Now that we've covered how traits are inherited, the final piece of the puzzle is understanding how genetic variation influences evolution and conservation. Inheritance isn't just about individual traits. It plays a crucial role in shaping populations over generations. This is where the principles of evolution by natural selection and conservation genetics comes in. Natural selection is the process by which individuals with beneficial traits are more likely to survive and then reproduce, passing those traits onto the next generation. Over time, this leads to evolutionary change in a population. The key driver of natural selection is genetic variation, which we now know comes from mutations, meiosis, and sexual reproduction. Here's how it works. Step 1 is that variation exists in a population. Not all individuals are genetically identical due to meiosis and mutations. Step 2 is that some traits provide an advantage in their respective environments. Individuals with certain characteristics, for example, a faster speed, better camouflage, or resistance to disease, have a better chance of surviving. Number 3 is that those individuals reproduce more. because they survive longer and can pass on their beneficial traits. Number 4 is that over generations, the frequency of advantageous traits increases while less beneficial traits become less common as they die out. A classic example is the peppered moth during the Industrial Revolution. Before pollution darkened tree bark, light-coloured moths were more common because they blended in with their environment. But once pollution covered trees in dark soot, these darker moths then had a survival advantage and became more common. This shift happened because of natural selection acting on pre-existing genetic variation. But what happens when genetic variation is lost? That's when conservation genetics comes in. Some populations, especially endangered species, experience genetic bottlenecks, where their numbers drop dramatically due to habitat destruction, disease, or hunting. When this happens, genetic diversity is drastically reduced, making the species more vulnerable to extinction. With fewer alleles in the gene pool, populations struggle to adapt to environmental changes, and harmful mutations become more common. A real-world example of this is the Tasmanian devil. A contagious cancer known as devil facial tumour disease, or DFTD, has devastated populations. Because Tasmanian devils already have low genetic diversity, many individuals lacked resistance to the disease, making it spread rapidly. Conservationists are now selectively breeding devils with more genetic variation to help the species recover. Now that we've broken down the key concepts in Module 5, you should be able to see how these ideas are all deeply interconnected. Rather than memorizing individual facts in isolation, the best way to approach Module 5 is by recognizing the bigger picture. DNA holds the instructions for life, and then those instructions are used to build proteins. Genetic variation arises through meiosis, and inheritance determines how traits are passed down. Over time, these inherited traits shape populations and influence evolution. If you can confidently connect different ideas across Module 5, you'll be in a much stronger position to tackle application-based exam questions, which is exactly what Band 6 students do well. Now that you've gone over these concepts, one of the best ways to consolidate your understanding is by creating a concept map or structured summary. Instead of treating each topic separately, try mapping out the relationships between mutations, gene expression, genetic technologies, ethical considerations, and biodiversity. Seeing these connections visually will help reinforce everything we've covered to make it easier to recall in the exam. If concept mapping isn't your thing, another effective strategy is to summarize each topic in your own words, and then challenge yourself with past HSC style questions. The key is to actively engage with the material, whether that's through diagrams, self-explanations, or practice questions, so that you can confidently apply your knowledge under exam conditions. The more you interact with these ideas, the stronger your understanding will be, and the more prepared you'll feel walking into your HSC Biology exam. And to help you with this, I recommend using Excel HSC Copilot, a study platform that includes concept maps and past HSC papers, with instant feedback on your answers using AI technology. Good luck and happy studying.